So now I'd like to introduce our first keynote presenter, David Keefe, who will be giving a talk on reproductive genomics.
So this is a very exciting time for reproductive genomics, and we're looking at microbiome, male infertility, female infertility, so many different areas. And in 20 minutes, I didn't know where to begin. So I followed Matthew 20, may the last be first and the first last, and many are called but few are chosen. I chose probably the least represented area of reproductive genomics, which is embryo biology, embryo genetics, pre-implantation genetic testing. Currently a challenging time to do research funded by National Institutes of Health because of the Dicker-Wiki Amendment, but at the same time a tremendous opportunity because now this is a clinical test. This is where the rubber meets the road. This is what clinicians are practicing every day all across the United States, and they're creating biobanks that are used clinically, which could be accessed, I believe, and this is potentially discussion for the bioethics people. Could we access the large resources now that are being collected clinically? So I chose to focus on pre-implantation genetic testing, understanding that the extensive breakthroughs in the other areas of male infertility, the genomics of female infertility, and microbiome will be picked up in the other discussion sessions. I'm got a couple of disclosures. I'm on the clinical experts panel of Illumina, scientific advisory boards of Origio, Cooper Genomics, and have funding from the March of Dimes. So I want to very quickly go through pre-implantation genetic testing, including PGD and PGS. Look at the profound impact of genomic instability on early embryo development and summarize past attempts to use genetic screening to improve reproductive outcomes of couples going through IVF, and report how recent improvements in pre-implantation genetic screening, or PGT-A, may improve outcomes in select patient populations, and then articulate what I believe are some of the urgent questions surrounding the clinical utility of PGS 2.0. This is actually in play now, widely used clinically, and then consider other contributors to genomic instability. Pre-implantation genetic screening, also known as pre-implantation genetic testing for aneuploidy, tests for chromosome copy number, typically to determine the chromosome status of the embryo, since this is a major driver of failed implantation and failed pregnancy since then. There's been an evolution of the field. Early attempts looked at the polar body, understanding that meiotic non-disjunction is a major driver of age-related infertility, age-related miscarriages. Early work by Berlinski, Handyside, and actually ongoing work by Handyside has shown there is some value of screening the polar body. First, they used fluorescence in situ hybridization, and more recently, comprehensive chromosome screening. Limitations of this are that there's a potential for correction. Yuval Yaron and others have shown there's a correction during mitotic phases of non-disjunction during meiotic phase, and then of course we now know there's extensive mitotic non-disjunction as well. PGS 1.0 involved blastomere biopsy, so this was cleavage stage, and typically early on evaluated selective numbers of chromosomes, those most affected using fluorescence in situ hybridization, and was actually shown to be worse than doing nothing, presumably because cleavage stage, you have 6 to 8 blastomeres. You're removing a substantial bulk of the embryonic mass at a time when there's extensive genomic instability and mosaicism, and fish had very limited ability to look at comprehensive chromosome screening. The current method, we call 2.0, involves trophectoderm biopsy, growing the embryo out to the 5th or 6th day, by which time the genomic instability has settled a bit. Mosaicism is less common, although still present. Allowed comprehensive chromosome screening with 4 to 6 cells being analyzed rather than just 1 or 2, with a much less invasive impact on the resulting on the biopsied embryo. And initially using array comparative genomic hybridization, and more recently next generation sequencing. A number of groups are looking at what some may call PGS 3.0, including non-invasive screening of blastocysts in culture using some of the cell-free DNA used in fetuses, but using them in preimplantation embryos, and others are starting to look at RNA, using single-cell RNA-seq as a way to look at copy number, but also looking at expression. Those are not widely used clinically, but are kind of in the pipeline. Just to drill down on the two sort of major stages that hit the clinical arena, as I mentioned, 1.0, the initial use cleavage stage, 5 to 8 cell embryos, PGS 2.0 looks at the blastocyst, which has many more cells, and they've already begun to differentiate, so you're biopsying from cells destined to become the placenta, chorion, and/or membranes. And the approach was typically on day 3 of a stimulated cycle. The newer method tends to take advantage of the extensive efficiencies now accrued through vitrification, cryopreservation, which allows transfer in an unstimulated cycle, which turned out not only to be advantageous to allow proper analysis of the biopsy specimens, but at the same time, it was actually abrogated much of the detrimental effects of controlled ovarian stimulation on the endometrium, including hypertension in the resulting pregnancy, during the resulting pregnancy, disrupted implantation. It appears that cryopreservation with transfer in an unstimulated cycle is actually advantageous, get more cells to biopsy, and the analysis of multiple chromosomes, primarily through array or next-gen, with a few groups still using a multiplex qPCR method, and you can look at all chromosomes. So the number of studies, again, with 20-minute time frame, I can't fully pay credit to all the groups that are looking at this, but this is one of several studies, happens to be from our group, in which you can see, SART stands for the Society of Assisted Reproductive Technology. These are the national statistics for pregnancy by age. You can see down at the bottom less than 35, 35 to 37. You can see this very progressive decline in pregnancy rate as a function of age. But if you look in purple and violet, these are embryos that had previously been screened, shown to be euploid, and you can see the effects of age are largely abrogated, at least in implantation. Now, obviously, not everyone gets to transfer in these older groups, and so begs the question, if you look at intention to treat analysis, would this be as advantageous? But this by itself is enormously valuable because a failed pregnancy following a very expensive IVF for many couples is a dread event, an expensive event, and should there be a miscarriage, it can drag the woman's attempts at fertility out for 4 to 6 months as the pregnancy resolves. So by blocking that, we're able to accelerate the whole process. There is a randomized trial that's looked at the efficacy of the PGS method using TE biopsy and next-gen. This was funded by Illumina. The primary outcome was pregnancy at 20 weeks, which we know is nearly delivery. And they looked at PGS in the experimental arm, and then standard selection, which is done through morphological analysis in the control. It was a multi-center trial with 34 sites, multiple laboratories in 4 different countries attempting to recapitulate the actual practice, 650 cycles, patients randomized. You can see the age groups from 25 to 40. They had less than or equal to 2 prior failed cycles and less than or equal to 1 miscarriage, and they were not carrying a major genetic Mendelian condition. And you can just see the overall schematic in which the IVF was carried out. There was retrieval, and then 5 to 6 embryos were checked, and then there was a randomization either to the control arm, which was a standard morphological assessment, versus the PGS arm using next-gen and comprehensive chromosome analysis with vitrification followed by a single embryo transfer and the outcome being 20 weeks. And you can see particularly in the group that is actually the biggest bulk of our patients, particularly in large cities, practice in New York City, you can see the 35 to 40-year-old group is not only the most common. Our average age at the New York University Fertility Center, which does about 1,800 cycles per year, is 37. So this is who we see. And you can see there was a significant improvement in the 20-week ongoing pregnancy rate. The younger group did not show this, and then overall, the net effect overall, when including all comers, did not show an effect. The miscarriage rates were surprisingly low. We ourselves do not see in this 35 to 40-year-old group an 11% miscarriage rate, and nobody knows quite what happened there, but clinical trials are always quite an adventure in terms of the way they get implemented. To us, this is very welcome to have a randomized clinical trial showing any effect of anything in our field of infertility. A lot of what we do ends up being empiric, done outside a conventional NIH-funded scenario. But the questions that are pending about this will intention to treat analysis, which randomizes patients before they start the cycle, show benefit. How do we manage these the tremendous variability when you're dealing with a procedure like pre-implantation genetic screening? The lab part is standardized, but the biopsy, the clinical management of the patients is very variable. And one factor which we'll deal with in a second, mosaicism, is of increasing importance clinically. The next gen is showing that there's a very high rate of mitotic non-disjunction. We're seeing mosaic embryos. This study was initiated before the urgency of the mosaicism question had really become obvious, so they were just essentially not included. And the other questions which are very pressing, urgent, are why, even when you're transferring a euploid embryo, almost half of the embryos still fail to transfer and or miscarry? Endometrial factors and other genomic factors. Mosaicism, as I said, is an urgent problem, one that really needs addressing clinically in basic science. As you know, it's two or more populations of cells with different genotypes in one individual. And it begs the question, is this mosaicism an artifact, a technical artifact of the technology itself? But regardless, we had a very high concordance rate for aneuploidy, 97%. And then a big discussion, especially following the work from Magdalena Zenica-Gertz in Cambridge, that there's a big difference, at least in mouse, between inner cell mass and trophectoderm in terms of the way they manage aneuploidy. We did not see discordance between the inner cell mass and the trophectoderm in human embryos. So we're pretty confident that what we're seeing is very reliable. Mosaicism, on the other hand, was a lot harder to get concordance. There was a significant disconnect within embryos between the whole genome sequencing in one versus the other piece, even if we're both looking at both trophectoderm and both. So we think that this is actually potentially not technical but biological, in which you have an embryo, and here you have euploid cells in brown and aneuploid in purple. And this would have been a sampling issue in which these came back euploid, and then in other cases you happen to sample this mosaicism in you'd call that aneuploid, and then you can imagine a scenario in which you get a mixture, and that we call that mosaicism. So this is, we believe, at least partly biological. Mosaicism is very likely real. We know that it's a time of tremendous genomic instability. This has actually challenged our approach to reproductive genetics early on, because most of us have come to assume that the result will be binary, normal or abnormal, and what we're getting instead is this mixture. And so we need to learn from the perinatal medicine, in which they look at risk factors and be able to do risk scoring. The second question I want to very quickly touch on is, why do we have nearly 50% of euploid embryos still failing? This is the same data, but you can also flip it around and look up there. What else is going on? And we know there's other things other than just chromosomal abnormalities. Copy number variants are ubiquitous. This is a study from the group in Louvain that shows very widespread copy number variants in human pre-implantation embryos. There's extensive evidence that especially during times of there's marked demethylation, epigenetic changes, that there's a tendency towards chromosome N fusions. This is the work of that comes out of Titchida Lang's group that shows that with the uncovering of the telomeric ends, you get formation of dicentric chromosomes, and you end up getting anaphase bridges that end in chromothripsis. We, for about 10 years, have been focusing on telomeres in early reproduction and shown that a mouse model with mouse have telomeres about 10 times longer than humans, and by genetically or pharmacologically shorten them, you can phenocopy much of the reproductive phenotype with humans. And then what we've moved in more recently is collaborating with Jeff Boca at NYU and Kathy Burns at Hopkins. We've been looking at the possibility of whether this marked demethylation, which is widely acknowledged, occurring following fertilization and activation, is leading to activation of some of the still active retrotransposons. And this is just very preliminary, but we have evidence that the same alternative lengthening of telomeres that we'd shown in mice happen in humans, that we get an expression of a telomeric RNA, using the telomere really as kind of a model to look at the more general question of retrotransposition and genomic activity. In the fly, you can see that this happens naturally during development, in which there's a marked expression of these RNAs that leads to expression of reverse transcriptases, endonucleases with insertion, and we've shown that inhibition of these will disrupt the telomeres. We see expression of some of the retrotransposon proteins, and we're working with Kathy and a guy called David Fenio to profile these. You can actually use a vector based PCR mechanism to preferentially amplify in sequence very densely where the still active line 1 insertions are actually, whether they're disrupting developmentally competent embryos. The idea being that a lot of what's going on is going to be outside of the exonic region, but it's such a vast landscape with such a challenging architecture that it's going to be hard. So to wrap it up, genomic instability is ubiquitous in pre-implantation embryos. It's a lot like cancer. Many have argued that the embryo copies cancer. I would argue that cancer usurps many of the mechanisms that probably developed during early embryo development. You get genomic instability, disruption in methylation, rearrangements, chromothripsis, and that's why you see mitotic as well as meiotic errors, mosaicism, copy number variants, and this DNA recombination-based telomere elongation. And so that's it. Thank you for your attention. Do we have time for questions? Or?
Thank you very much. I think I will be moderating from here the way we have the room set up now. We do have a few minutes for questions. Please remember to speak into the microphone.
We have a question on the phone from Gwendolyn Quinn. To what extent do you think patients understand this information? Okay, great. We have a question from Gwendolyn Quinn on the phone, and she says, to what extent do you think patients understand this information and their odds of success of a healthy baby? In your experience, even in the face of poor odds, are patients starting to continue?
That's a great question. The question of the patient's perception of genomics in general, but specifically of reproductive genomics, and most particularly in genetic testing. Before I was an OB/GYN, I was a psychiatrist, so I spent 6 years trying to understand what people understand. Hope springs eternal. And one of the hazards, especially in the U.S. system, is that you have, a capitalistic, essentially entrepreneurial healthcare system delivering this in the vacuum without a lot of funding from NIH, without a lot of oversight. And so there's a bit of a conflict of interest where patients can be led down a path where they're hearing what they want to hear, and it's leading to potentially gaps. But it's very important. How does I think it begs for the question: could other sources resolve NICHD, the Genome Center, provide materials so that people could get a little bit more objective, disinterested information about the probabilities. But I had a patient come to me just a couple of years ago who told me she's going to continue to do IVF with PGS until she has a child. She's 45 years old, and in our program, I can think in one hand the number of people that actually achieve pregnancy. And she just continued to do IVF with PGS until she got a euploid embryo, transferred it, and has a baby girl. So it cost her $275,000, which as a finance person in New York, she said, that's not even, a couple of months bonus, which, that's not scalable, obviously. And who decides, is it worth it for an investment banker to have one more child versus access to the masses? So these are very, very important questions about access, information, who provides it, who controls it, and a great question.
Given the very striking graphic you showed of the mosaicism in the embryo, it seems to me like one could also say that physicians and companies have been overly hopeful that this screening is actually a reliable test, and given the very high false positive rates that it has, and it's been sold to patients for years now as a determinative test that they could rely on, and probably we know that viable embryos have been discarded and people have failed to achieve pregnancy. So can you just reflect a little bit on what we've learned from this test, which is a test that was sold before it was ready and before it was thoroughly tested out and did?
Right. Well, I didn't take any selling of this test. The patients dread a miscarriage. They dread a failure. So they seek it. But the more fundamental question is, does it provide value or not? If you think of it as a diagnostic test, no, it's not a diagnostic test. If you think of it as a screening test, yes, I do believe it provides substantial value. For example, NIPT, is it 100%? Are there examples where it failed? Obviously, this is more like that. This allows us to rack and stack the embryos. Now, you're right, there has been misuse of this test. For example, some centers are very risk averse, especially in New York City. The risk of a baby that turns out not to be perfect and then coming back and it was because you transferred my mosaic embryo. We spend a lot of time counseling. We actually will transfer mosaic embryos. There's a publication that has come out that helps us prioritize them. Some we will typically recommend not be transferred if your mosaic and includes a viable aneuploidy like 21, 18, 13, or includes chromosomes that are mosaic chromosomes that are at risk for uniparental disomy. We would not transfer those mosaic embryos. But a lot of the mosaic embryos appear to self-correct. The way I explained, I think one of my patients was the first that went through transferring a mosaic embryo because that's all she had left. And we explained that 2 years ago when we were using array, we wouldn't have known about this mosaicism result. So as a result, we will use it as a screen, and we'll use it to counsel about relative advantage or disadvantage. I suspect if you never transfer any mosaic embryo, you'll probably never, you'll have a lower success rates overall, which, it's a great point. It's depending on how you use it. It's information, it's neutral, but it can be used to advance or to delay the progress of the patient. I think using selective embryo transfer, using the mosaicism call to maybe delegate the mosaic embryo to a lower position and we actually have a study of our mosaic embryo transfers, and you get pregnancies with about half the implantation rate and twice the miscarriage rate. So you still can get pregnant. That's what we use to justify. The fear everyone has is what happens if this moves towards the placenta and you end up getting intrauterine growth retardation and the child is affected, or there's some mosaicism in the body that continues and creates some congenital anomalies, and this may happen. And we spend a lot of time going over that. And again, you can imagine a scenario where you have private practices that are based on volume. Everything is based on volume. So this is just information that's annoying. And there are actually a number of the genetic screening companies that their market position is not to provide the information. And that, for some clinicians, is an advantage. They don't get the information. They don't have to talk about it. But I think overall, if you look at the value, if you can prevent a miscarriage in somebody that's losing fertility at a rate of 1% per month, if you can prevent a miscarriage, then you potentially could keep her in the game while she still has a chance, rather than having her have a miscarriage by transferring, say, a mosaic embryo that had a very high chance of miscarriage, or aneuploid embryo that you didn't know about, then you end up, she can go from being 40 where she has 20% chance, and then she could be out of the game by the time she's out of the window there where she's pregnant and miscarrying. So.
But we do have time for a few more. Dr. Geese, Dr. Gray, I've seen Dr. Morton's. There's a question here. I don't want to be ignoring people in the back of the room, so you may have, I see one hand going up. Hands may need to go up as we learn how this is working during the day. But I'll start with Dr. Jadis.
I don't mean to waste there we are. David, thank you for a very comprehensive overview. I'd like to hear your thoughts, please, about PGS 3.0, the cell-free or non-invasive approach, because of the granted mixed data for safety of embryo biopsy with regard to increased rates of low birth weight, and also the controversial area in IVF of freeze-only or freeze-all with regard to large birth weight. So if one can bypass the actual biopsy of an embryo and freezing, looking ahead to PGS 3.0.
Exciting concept, still guarded. The advantages are all you mentioned. Potentially, one of the reasons we don't see an advantage in the STAR trial in the early group may be that there is a disruptive effect, even in experienced programs. I think our embryologist was on the first paper in 1991 when she was still at Cornell with Jamie and Sav. So she's experienced, but nobody's perfect. It's a procedure, and if you could avoid that, that'd be huge. The flip side of it, though, is I'm still trying to get my mind around it. I've read Sunny Shee's paper. The Brigham and women people are pioneering this work. A number of other groups. I know you're doing some in California, UCSF. Some Chinese groups are working on it. I have questions. So the models that I've seen, and maybe you can help me with this, they take an embryo that's already been biopsied, so they have the call on its normalcy. And then they collect the media. And the question is, do you need that breach in the zona? The zona is a loose mesh of these intermediate filaments, CP1, 2, and 3, but they're highly charged in how these fragments, these 300 meters or wherever big they are getting through the zona in the absence of a hole that's already been there. I just wonder, is it going to be as helpful, number one? Number two, is it going to exacerbate the mosaicism question? Because presumably the apoptotic cells that are preferentially contributing to the culture media cell-free fetal DNA must be different in some way. Are they more likely to be aneuploid? In other words, there's going to be a sensitive but less specific test, or so those are the questions. The breakthroughs have been Sunny Shee's application of some more linear approaches to amplification, the Malbach, and so his new method, which required for that, it remains to be seen. If we can do what they're looking like they can do, it'll be very valuable, and I could see it being applied to all IVF without the risk. The flip side of it, though, it's going to bring on that same question as the specificity of the diagnosis or the call on the cells, and whether that's accurate of what's going on in the inner cell mass in the future embryo. So.
Yeah, Tony Gregg.
We are down to 0 minutes.
Okay.
Flashcards up
Okay.
there. Do you have a question that can be answered in 30 seconds or less?
I have just very quickly, I'm going to just put out 3 questions. You don't have to answer them now, but
Yeah.
are we misinforming patients and the public by calling these embryo biopsies? Just a question, because it's not the inner cell mass. Number 2, the concept that we're picking up mosaicism of whole chromosomes that have not yet reached a z-score of 2 or greater. There's a lot of other across the genome blips that are occurring. Are we ignoring significant sensitive information by ignoring everything except those that reach a z-score of 2? And number 3, are we oversimplifying the trophectoderm? In other words, it's a 3-dimensional structure, and does it matter where the biopsy takes place? I'll just leave those questions on the floor.
Yeah, and we'll have plenty of time,
Great,
I think, in
thank
the
you.
working groups.
Yeah, that'll be great for the
Yeah.
working discussions that we have, particularly tomorrow. So I'm afraid I need to close down this presentation. Please, everybody who had questions, write them down, remember them, again, so we can have other opportunities to continue to explore this topic as the conference goes on.
Great, thank you, Lisa. Yes, we'll have an opportunity during the breakout sessions today and tomorrow, and discussions this afternoon.